technology. Time permitting, we will conclude today's webinar with a brief question and answer session. If you have a question for Dr. Kubler during the webinar, please use the questions feature located on the GoToWebinar control panel on the right side of your screen. The control panel is automatically collapsed upon opening the program, so if you don't see it, please look in the upper right corner of your screen for an orange arrow button and select it. This will open your control panel where you'll be able to view the questions feature. Please type your message or question in the box and press send. Today we will hear from Dr. Sigrid Kubler, who's been the Director of Customer Service at Wyatt Technology since 2012. She's been directing and teaching Wyatt's Light Scattering University program for many years. Before joining Wyatt, she worked at Symix Technologies, where she developed various high-throughput screening techniques for materials characterization. She studied chemistry at the University of Mainz and the University of Toronto, with her doctoral work in the area of polymer structure and dynamics conducted at the Max Planck Institute for Polymer Research. So without further ado, please allow me to welcome Dr. Sigrid Kubler.
Thank you for the introduction, Lindsay, and welcome everyone to Light Scattering 102, Principal Applications of Light Scattering. And welcome back everyone who has already attended Light Scattering 101, Fundamentals of Light Scattering. This webinar discusses some of the primary applications and uses of multi-angle light scattering and dynamic light scattering. In today's webinar, we will first go through a quick review of the light scattering fundamentals we've discussed in depth in the first part of this webinar series. We will then discuss the fundamentals of using light scattering with separation techniques, in particular size exclusion chromatography with light scattering detection and dynamic light scattering detection. Examples will be used to illustrate what we can learn from SEC-MALS and SEC-MALS DLS, as we call the techniques. Asymmetric field flow fractionation, in short AF4 or FFF, is an orthogonal technique to size exclusion chromatography, and we will discuss fundamentals and applications of this highly versatile separation method when used with light scattering. Lastly, we will discuss batch light scattering techniques and their applications. What can light scattering measure? As we have seen in Light Scattering 101, there are several categories of light scattering techniques. The first one, namely static light scattering, measures the absolute intensity of scattered light independent of time, and is also often called classical or Rayleigh scattering. Fundamental molecular properties, such as molar mass m, RMS radius, or sometimes called radius of gyration, and the second virial coefficient A2 may be determined with Rayleigh scattering. The second technique is dynamic light scattering. In DLS, we observe the rapid fluctuations of the scattered light that originates from Brownian motion of molecules. Dynamic light scattering measures a translational diffusion coefficient of a solute, from which a hydrodynamic radius may be determined. We will not discuss the third technique, phase analysis light scattering, or PALS, in this webinar. In short, PALS measures the electrophoretic mobility of a molecule and determines charge of a molecule in an external electrical field using laser light scattering. Here I like to summarize the essential concepts of light scattering that have been introduced previously. First, the absolute light scattering intensity is proportional to the molar mass m, the mass concentration c, and the square of the refractive index increment dN over dC. The refractive index increment is sample specific, and it describes the change of refractive index of a solution when adding a solute. and it can also be viewed as a light scattering contrast parameter. In light scattering, dN over dc is the equivalent of the UV extinction coefficient in UV vis. Second, the angular dependence of scattered light is proportional to the average size of the molecule, more precisely the RMS radius of the scattering molecules. For light at the wavelength of 660 nanometers, we can distinguish two scenarios. Small particles with a radius smaller than 10 nanometers scatter isotropically, that is, they scatter the same amount of light in all directions. For larger particles, more light will be scattered at the smaller angles or forward direction. Less light will be scattered at, for example, 90 degrees or the larger or backscattering angles. The third concept describes the fundamentals of dynamic light scattering. Rapid light scattering fluctuations due to Brownian motion can be related to the diffusion coefficient and subsequently to the hydrodynamic size of the scattering molecules. Light scattering may be practiced in two operation modes. In chromatography, we are able to separate a polydispersed sample and we can assess molar mass and RMS radius moments, fractions, and distributions when using static light scattering. When using dynamic light scattering, the resolution and hydrodynamic radius is determined by the resolution of the chromatography separation itself. In a batch experiment, we are measuring an unfractionated sample, and when using static light scattering, only the weight average molar mass MW, z-averaged radius Rz, and potentially the second virial coefficient A2 may be determined. No information about the polydispersity of the sample can be obtained from static light scattering. When using dynamic light scattering in batch, we are able to obtain limited information on polydispersity by using deconvolution algorithms and software. We will first discuss light scattering in chromatography mode. Light scattering can be readily coupled to liquid chromatography and is a natural match for measuring molar mass of macromolecules. It is most often practiced in conjunction with size exclusion chromatography, SEC, also often called gel filtration chromatography. When used for polymers, in particular in an organic mobile phase, SEC is also often termed GPC or gel permeation chromatography. SEC separates molecules based on their hydrodynamic size or volume. Other chromatography techniques that may be used with light scattering are, for example, reversed phase chromatography, hydrophobic interaction chromatography, or ion exchange chromatography. But size exclusion chromatography is by far the most commonly techniques used with light scattering. In order to understand why multi-angle light scattering is so advantageous when using SEC, we need to look at the separation mechanism. Size exclusion chromatography separates molecules according to their hydrodynamic volume in a porous matrix. As the solute moves along with the solvent through the column, smaller solutes, like the one shown in red, can diffuse in and out of the pores of the matrix. Larger solute molecules, such as the blue or purple, will move faster through the column because they can penetrate the pores less. As a result, larger molecules will elute first, as shown in the chromatogram below. We're actually not primarily interested in the hydrodynamic size, but rather molar mass of a macromolecule. In SEC, column calibration, as shown on the right, is employed to relate the elution volume of the solute to its molar mass or molar mass distribution. Size exclusion chromatography is a relative method since it requires calibration with molecular standards that have the same elution behavior as the sample of interest. Coupling SEC to multi-angle light scattering, however, overcomes this limitation. Since light scattering measures the absolute molar mass, we can now determine absolute molar mass independently from column calibration. We may also change the HPLC setup, such as solvents, column configuration, detectors, without the need to recalibrate. Macromolecular characterization falls into two general categories. The molar mass distribution for samples with discrete species, such as proteins or biological samples, generally follows a step-type pattern due to the presence of monomers, dimers, trimers, or maybe even higher oligomers. When characterizing these samples, we are interested in detecting, identifying, and quantifying molar mass, aggregates, and fragments. Additionally, the chromatogram shown below nicely demonstrates the different sensitivities of the concentration detector and the light scattering detector. Whereas the refractive index detector is sensitive to concentration only, the light scattering detector is sensitive to both the molar mass and concentration. Now, when the light scattering and RI signals are normalized for the monomer, we can see twice the light scattering intensity for the dimer as compared to the refractive index signal of the dimer. The molar mass distribution of a polydispersed sample, such as the dextrane shown on the right, is generally continuous. We are primarily interested in determining molar mass moments such as number average, weight average, and z-average, and the polydispersity of the polymer. Note that here the different detector sensitivities manifest themselves in the relatively higher light scattering intensity at the higher molar mass fractions that elute first. What are the challenges with column calibration? In this example, a synthetic polymer with a wide molar mass distribution shown in the blue chromatogram was measured and compared to a linear 200 kilodalton and 30 kilodalton polystyrenes shown in red. The overlay of the hydrodynamic radius for both the polystyrene and the unknown polymer are the same for a given elution volume. This confirms that the sample is separated by hydrodynamic volume and column interactions are largely absent. The chromatogram on the bottom shows the overlay of molar mass versus time. Here we can clearly see that the molar mass of the unknown polymer is higher at the elution volume of the 200 kilodalton polystyrene standard. Higher molar mass for a given size indicates a more compact architecture. For synthetic polymers or biopolymers, smaller size for a given molar mass is often caused by branching. For biological molecules, a change in conformation may be caused by conjugation or binding or unfolding, and the conformation for these molecules is often unknown. Let's take a look at the experimental multi-angle light scattering setup with size exclusion chromatography. The HPLC itself consists of a solvent reservoir, degasser, HPLC pump. We often add an inline filter before the sample injection to remove any particulates that might be introduced into the separation system in order to obtain the highest quality light scattering data. Samples are injected via an autosampler or manual injector and then separated by a column or a set of columns. After the separation, the elute flows through a series of detectors. Shown here are UV-Vis, multi-angle light scattering, and differential refractive index detectors. The signals from each detectors are transmitted to the chromatography software and analyzed. Note that this particular setup contains two concentration detectors, a UV-Vis and a DRI. A second concentration detector may be used to confirm the measured concentrations, compute sample or detector response, or analyze protein conjugates or copolymers. Before we can analyze our samples, we need to consider two effects when multiple detectors are used. The first one is called inter-detector delay and must be corrected by alignment. After the sample has been separated by a column, it will pass through the series of detectors as we've seen on the first slide. The UV detector response is shown in green, light scattering response in red, and DRI detector response in blue. We need to determine the physical delay volume between these three detectors, since in actuality we want to determine the concentration and light scattering signals at the same slice to calculate accurate molar mass. Software is used to overlay the signals to correct for that physical volume between multiple detectors in series. We do need to use a monodisperse sample to perform alignment to account for the different detector sensitivities of the light scattering and the concentration detectors. For example, bovine serum albumin or a low molecular weight monodisperse polystyrene standard. A second effect of detectors plumbed in series is band broadening, that is, the additional peak dilution as a sample flows through more than one detector. Shown to the right are the traces for the light scattering detector in red and the DRI detector shown in blue. One can clearly see the effect of sample dilution at the tail end of the peak where the RI trace is much broader. This results in a curved molar mass trace as shown in black, even if the peaks in the chromatogram are in fact monodisperse. This interdetector band broadening must be corrected by the chromatography software. After band broadening correction, the traces of the monomer peak overlay as expected, and the resulting molar mass distribution is monodisperse. Note that as for determining the interdetector delay volume, a monodisperse sample must be used for the band broadening correction. This is often done in the same experimental run. And as before, in many cases BSA works well for aqueous chromatography, and a 30 or 50 kilodalton polystyrene standard works well for organic chromatography. We've now discussed all the fundamentals and experimental considerations of SEC mols and are now ready to take a look at some examples. This chromatogram shows hepatitis C virus NS3 helicase protein, which catalyzes DNA unwinding. We can identify the monomer with approximately 48 kilodalton, a dimer, a tetramer, as well as fragments. Light scattering can give precise information on molar mass of aggregates, even if they are present in very low amounts. The aggregate and fragment peaks are shown in more detail in the inset, whereas in the chromatogram itself, they are actually of very low intensity. Here, the RI detector was used as a concentration detector, and a particular advantage of RI detection in this example is that the response is near constant for proteins, since the variations in dN over dC for different amino acids are averaged out in the protein. This makes the RI detector a very versatile concentration detector, since it is not necessary to determine the UV extinction coefficient in order to calculate concentration. In fact, we may use the DRI response to calculate the UV extinction coefficient of the protein monomer as shown below. SEC molds may be used to determine stoichiometry of complexes, such as binding the helicase protein with a small DNA molecule. The chromatogram in red shows the results of the complexation, while the chromatogram in green shows the original protein molecular weight distribution. Light scattering determines a molar mass of approximately 54 kilodalton, which confirms a one-to-one stoichiometry of the protein DNA complex. Also visible is a small residual fraction of unbound DNA. For polydispersed sample, cumulative molar mass distributions are an excellent way to compare results and can help visualize difference between polymer samples. This example shows different ranges of molar mass distributions for 3 hyaluronic acid samples, which is a biopolymer with high molar mass and a wide molar mass distribution that is widely distributed throughout tissues. When comparing the RMS radius distributions, one can see that the sample labeled hyaluronic acid 1 is highly compact as compared to sample 3. Its RMS radius distribution is very low. A graph of RMS radius versus molar mass for polydispersed polymer is called a conformation plot, since a conformation plot indicates the compactness or conformation of a sample. We can see in this example that samples 2 and 3 have different molar mass ranges, but they have the same conformation since they have the same slope. The measured slope of approximately 0.5 indicates the conformation of a random coil polymer. In sample 1, the conformation plot slope of 0.18 points to a highly compact conformation, and in our case, this compact architecture is due to cross-linking. Below the graph, conformation plot slopes for different sample architectures are shown, such as the slope for rods, random coil polymers, or spheres. This chromatogram of a protein nucleosome complex illustrates how the various light scattering techniques can help us understand the elution behavior of a sample. Size exclusion chromatography identifies 3 distinct species, which would as first indicate the presence of dimer or higher oligomers. However, light scattering measures essentially the same molar mass for all species. Dynamic light scattering can provide more insight into the size of the different species present. In this protein nucleosome complex, dynamic light scattering reveals a hydrodynamic size increase for the second peak in the chromatogram, which indicates a partial unfolding of the complex and simultaneous presence of multiple conformations of the same molecule. Note that if we had only looked at the results from DLS or the chromatogram without any static light scattering data, we would have likely and incorrectly concluded that our complex forms aggregates such as dimers and trimers. Had we only looked at the molecular weight, we would not have been able to confirm the presence of different conformations or sizes. So the use of both DLS and MALS allows us to correctly identify conformation and helps explain the elution behavior of this complex. This example shows the benefit of adding multi-angle light scattering to SEC if the elution behavior is not ideal. The chromatogram in red shows a generation 5 polyamidoamine dendrimer with the broad and asymmetric peak shape indicating the presence of column interactions. Despite the non-ideal chromatography, we are able to determine the correct molar mass and confirm that the sample is monodisperse as expected. The sample shown in purple is a G5 dendrimer prepared by a different synthesis route. The chromatogram and the molar mass appear highly polydisperse, and we can clearly see that this has a different molar mass than the standard dendromer for a given elution volume. This indicates either column interaction, different architecture, aggregation, or a combination of these effects. More insight into the behavior of these dendromers may be obtained by determining the hydrodynamic radius by DLS. From the chromatogram, shown in purple, it can be seen that both aggregation and non-ideal elution are present, since the molar masses are different for given elution volumes when compared to the standard. Lastly, batch dynamic light scattering confirms the expected size in the standard dendromer and aggregation in the sample prepared by the novel synthesis route, even without subjecting the sample to chromatography and potential aggregation or degradation. In the previous example, we have seen the limitations of size exclusion chromatography and interaction of a complex sample with a stationary column matrix. Asymmetric flow field flow fractionation is an alternative separation technique to size exclusion chromatography. Separation mechanisms for both techniques is the hydrodynamic volume of the molecules of interest. Conceptually, the HPLC setup is closely related to SEC with an FFF separation channel or a hollow fiber, replacing the SEC columns. For clarity, the FFF separation module that controls the various flows in the channel is not shown on the schematic of the slide. How is the separation by hydrodynamic volume achieved in FFF? Separation is performed in the liquid zone of several hundred micron height, and this is contained in a channel with a semi-permeable membrane lining the bottom surface. The laminar flow of the carrier fluid traveling from the inlet to the outlet creates a parabolic flow profile. A perpendicular cross-flow is applied to the laminar stream, which causes the analytes to accumulate in a small zone close to the membrane surface at the bottom. This concentration buildup on the membrane causes lateral diffusion back up into the channel. And since smaller molecules have a higher diffusion coefficient, they will be distributed higher into the channel where the carriage fluid is faster. Consequently, smaller molecules with a higher diffusion coefficient will elute faster than larger molecules with a lower diffusion coefficient. The elution profile in FFF is thus the inverse of SEC. What are the advantages of FFF? It's a gentle separation technique that can be used orthogonal to SEC. The lower surface area of the membrane also minimizes surface interactions. In field flow fractionation, the separation range is tunable by the strength of the crossflow. Samples of wide size ranges may be separated in a single run. A comparison of the molar mass distribution of a polypeptide conjugate from both SEC and FFF is presented here. The molar mass in this molecule varies from 1 million Dalton to over 100 million Dalton. And we can see in the SEC chromatogram in red that the molar mass is decreasing with elution volume as expected, but increasing at the tail region. This increase is likely caused by the so-called anchoring effect in SEC, which causes branched higher molar mass fractions of a polymer to be retained abnormally long in column pores and be co-eluted together with the low molar mass fractions. This anchoring phenomenon is not seen in FFF, where the column matrix is absent. Results from 2 injections shown in blue demonstrate the excellent reproducibility of the FFF method. Additionally, the weighted average molar mass from FFF is higher than that from SEC, suggesting a possible retention or shearing degradation In this conjugated example, we can see two potential FFF advantages. FFF is lacking the anchoring effect and has a wide separation range with minimum shearing degradation. Complex biological samples may be also analyzed by FFF. This fractogram shows a human serum sample from a patient. Serum albumins and immunoglobulins may be identified by their molar masses. Simultaneously, the hydrodynamic radius of the cholesterol particles may be determined. Measuring the size of, for example, low-density lipoproteins of interest, since it has been related to the risk of coronary heart disease. Combining size information obtained from MALS with size information from DLS allows us to learn more about colloidal structure. The fractograms to the left display the DLS radius and RMS radius distribution for filled and unfilled liposomes. One can see on the top left that the hydrodynamic radius measures the shell and remains the same, irrespective of the degree of filling. The RMS radius measured by MALS corresponds to the mass averaged radius of the liposome. The RMS radius will be smaller for a filled liposome for which the mass is distributed throughout the entire liposome rather than being concentrated on the surface. The ratio of the RMS radius over the hydrodynamic radius can give insight into the shape of the particle. To the right, we've displayed the ratio versus the elution time for both the filled and unfilled liposome. A ratio of 0.7 to 0.8 is indicative of a filled sphere, whereas a ratio of approximately 1 confirms the hollow structure of the unfilled liposome. We have seen that separation techniques yield detailed information about molar mass and size distributions. Sometimes it may be advantageous or simply faster to perform a measurement in batch. First, we will look at the classical batch multi-angle light scattering experiment, a Zimplot. Weight average molar mass, z-average RMS radius, and second virial coefficient may be determined in a Zimplot. Several known concentrations of an unfractionated sample are injected into a multi-angle light scattering detector, and the light scattering response is measured as shown in the inset on the top right. The angular dependence of the light scattering intensity can be related to the radius for the various concentrations. The concentration dependence can be related to the second virial coefficient. Molar mass is obtained from the intercept of angular and concentration dependence. Dynamic light scattering is a technique that naturally lends itself to batch measurements since it's quick and easy to perform and it does not require knowledge of concentration or the nature of the sample. We may even obtain some information about the polydispersity of the sample. A monomodal size distribution, as shown here, may be analyzed in terms of the mean and standard deviation of diffusion coefficients or radius. This is called cumulance analysis. This analysis requires that the sample of interest only has a limited polydispersity, for example, a protein monomer and its oligomers, or a polydispersed nanoparticle. Multimodal size distributions consist of several species with vastly different sizes. For example, small proteins containing aggregates in the hundreds of nanometer size range. Multimodal size distributions may be analyzed by regularization, a method to fit a set of diffusion coefficients and their distributions to find the underlying distribution of the hydrodynamic radius. An example of that is shown on the next slide. Here we have the overlay of monomodal and multimodal size distributions for a monoclonal antibody in two different formulation buffers, PBS and a buffer containing 2% sucrose. Each buffer formulation was measured for pre-lyophilized and lyophilized conditions. The lyophilized samples contain aggregates of various sizes, whereas pre-lyophilized samples are aggregate-free. We can display the presence of aggregates in terms of their light scattering intensity shown in the left regularization graph. This will emphasize aggregates. Alternatively, we can display the size distributions in terms of concentration or their percent mass, as shown on the right. We can see that aggregates contribute a significant amount to the light scattering intensity, approximately 20 to 30%, but only contribute 1 to 1.5% as a mass percentage of the sample. This comparison of the percent intensity and the percent mass distributions demonstrates the high sensitivity of batch DLS to the presence of a small amount of aggregates. What happens when we analyze the DLS data using cumulants analysis? Note that when using cumulants, we're not able to resolve different species. Therefore, the average hydrodynamic radii are shifted to larger values for the formulations containing aggregates, essentially averaging the protein and the aggregate peaks. Cumulants analysis will never resolve multiple species. Dynamic light scattering has been extensively used alongside differential scanning calorimetry and cycloidal dichroism to determine protein melting. The graph shows a plot of the radius versus temperature of an antibody formulation in two replicates. The size of the antibody protein remains constant until around 52 degrees Celsius, where we start seeing a rapid increase of the size. Since DLS only tracks a size change, DLS is both sensitive to protein unfolding and subsequent aggregation due to the exposure of the hydrophobic domains. It is therefore advantageous to describe the transition temperature in terms of an onset temperature, that is, the temperature at which this original size change is observed. Alternatively, if a stable final or even intermediate aggregation state can be reached, the transition temperature may be determined in terms of the midpoint as the transition, as shown in the graph to the right. When using the DLS plate reader, multiple formulations may be readily screened for their melting temperatures in a single thermal scan. Lastly, dynamic light scattering is capable of monitoring subtle size changes, as shown here for RNA molecules in various folding states. We can see a steady increase in radius as the RNA sampler investigated in its native, partially unfolded, or completely unfolded state. Since multi-angle light scattering cannot determine the RMS radius below 10 nanometers, small-angle X-ray scattering was used to determine the RMS radius, or RG. The increasing ratio of the radius from SAX and DLS confirms the transition from a compact folded molecule to an extended and unfolded conformation. At this point, I'd like to conclude with a brief summary of the light scattering techniques and applications presented in this webinar. We have seen that MALS and DLS are powerful tools that can be used in batch or with a separation technique. We can determine absolute molar mass and radius without using any molecular calibration standards and quantify the molar mass of distributions or fractions. Conjugation and stoichiometry of complexes may be determined by comparing molar mass and radius, or even just the radii obtained from MALS and DLS. We may learn more about conformation, branching, and molecular shape. Batch light scattering experiments will give us more insight into thermal behavior, conformation changes, interactions, and the presence of aggregates. When using both MALS and DLS, light scattering can aid with chromatography method development by comparing molar mass and size measurements to the elution volume. For more information, I invite you to go to our website and explore the various resources. For example, our application notes, webinars, and bibliography. You may also want to consider attending one of our training classes or user meetings. Thank you all for listening.
Thank you very much, Dr. Kubler. And to conclude the webinar, we will now have a brief question and answer session. Okay, first question. Can polyethylene oxide be used as a standard for detector band broadening instead of BSA or PS?
As long as the polyethylene oxide standard is of low polydispersity, this is an alternative. Monodispersity is generally defined as having a polydispersity index of less than 1.05.
Okay, and is light scattering a good technique to determine changes in molar mass with time?
It is an excellent technique to determine molar mass changes with time. In this case, most likely a batch experiment would be employed. Molar mass changes can be tracked directly by static light scattering and indirectly by the size change in dynamic light scattering.
Would you recommend FFF as an alternative to typical SEC columns for separating a mixture of synthetic polymers with molecular weights ranging from several thousand Daltons up to several 100,000 Daltons?
It can be certainly used as an alternative technique to SEC, even for synthetic polymers. FFF can be used with organic solvents. It has a particular advantage if the sample of interest does not elute well from a column.
Okay, what types of samples is the FFF technique most suitable for?
Generally, we use FFF for larger samples since we're limited in the upper size range. In size exclusion chromatography or another column chromatography technique. It is used as a gentle separation technique for samples that would be degraded or sheared by a column, samples that have column interactions. In short, essentially its main advantage is being used when SEC separation isn't possible.
And can absorbing samples be measured?
Yes, absorbing samples can be measured with light scattering. If the absorbance is small, we can actually correct by software and by measuring the absorbance in the light scattering flow cell. That is possible. If absorbance or fluorescence is too severe, one needs to measure at a different wavelength, which is also an option.
Can DLS be used to analyze biological sample in serum?
Yes, dynamic light scattering can be used for that. It depends also on the mixture, how complex the sample is. One gets limited resolution in batch DLS, that is to resolve separate peaks. One typically needs a size difference of 3 to 5. If that is not the case, it's actually better to use DLS for separations.
And what type of information regarding branching can be obtained from a conformation plot?
The conformation plot indicates the radius for a given molecular weight, and the slope can give an average of the type of extendedness or compactness of the molecule. Conformation plots of typically around 0.5 to 0.6 indicate random coils. The higher the conformation plot slope, the more extended the structure is. The smaller the conformation plot slope, the more compact the structure is. So it's a relative comparison.
And can light scattering be used for particle size measurement? And what is the size range?
Light scattering, typical light scattering size ranges when measuring DLS in batch are between 0.5 nanometer in radius and 1,000 nanometers in radius. When used with chromatography, due to the flow conditions, the size range is generally a little bit smaller. In DLS, typically up to 50, 150 nanometers, depending on the detection angle. In terms of RMS radius measurements, these are typically in the range of 10 nanometers, sorry, up to 500 nanometers.
And what information can you obtain from characterizing micelles in solution?
One can obtain from characterizing micelles in solution from a concentration-dependent measurement. One can determine critical micelle concentration above the CMC. One can determine the micellar size or molar mass even.
When would you measure a Zim plot and not use SEC mols?
A Zim plot is typically used when SEC mols is challenging. For example, there's no suitable column available, but the determination of molar mass and radius is of interest. It's also of interest if one wants to measure the second Rayleigh coefficient or monitor a time-dependent or temperature-dependent equilibrium or reaction.
And can you analyze a mixture of highly aggregated nanoparticles?
That is possible. It depends on, again, on the size distribution, which technique is most suitable for that. If a detailed size distribution is of interest, field flow fractionation would be the method of choice.
And what are the limitations of DLS and our SLS for high concentration protein formulations?
Limitations typically for high concentration formulations, in particular in DLS, are viscosity effects as well as electrostatic interactions. These interactions cause a change in the hydrodynamic behavior and the diffusion coefficient. So we typically see an acceleration of diffusion due to electrostatic interaction. We see, since diffusion is dependent on viscosity, we actually see slower diffusion at higher viscosities.
Okay, when measuring a polymer mixture, what DNDC value should be used for its measurements?
When measuring a mixture, I mean, one would, if only the average molar mass is of interest, one can either measure the DN over DC for that mixture, or more precisely copolymer, or one can use what we call the protein conjugate or copolymer analysis, where one uses both an RI and a UV detector, and the UV detector being differently sensitive to different components to determine the fractions itself and the total molar mass of the copolymer.
Okay, when we change the membrane of the AF4 channel, do we need to align and band broaden again?
Alignment and band broadening are effects between the various detectors, that is UV, light scattering, and RI, and as such, changing a membrane or a column configuration does not have any influence on that.
Okay, I think we just have one more question. Would it be sufficient to measure size only by DLS when using SEC?
That will only confirm or show the presence of column interaction or the absence of column interaction. It will not determine the absolute molar mass.
All right, well, we are almost out of time and must conclude our webinar today. Wyatt's on-demand webinar library is a great resource where you can find recordings covering many additional aspects of light scattering technology and applications. If you have further questions, please get in touch with me at the contact information in your webinar confirmation email. Thank you again, everyone, for joining us, and a big thank you to Dr. Kubler for the presentation.